So today we are discussing the counter-current multiplier. All right, the idea behind the counter-current multiplier is that as you dip down into the medulla, you go from an area of low osmolality or osmolarity, which just means salt content, to an area of high osmolarity, or salt content. So basically the deep inside of the medulla is going to be very salty. And there's a gradient that is established between the cortex and the medulla. And we're going to talk about exactly how that gradient is established. The numbers are this. There's only a few numbers you really need to know. In the cortex, it is 300 milliosmoles, which is just a measurement of the concentration of salts. You can basically just think of it as salt, sodium chloride, because that is the main contributor to this saltiness. And then as you dip into the medulla, you'll notice that the numbers change. As you go down the renal column, you'll have 400, 600, 900, 1200. And so deepest inside of the medulla, you're going to have 1200 milliosmoles, so very, very salty. So we're going to talk about exactly how that is established. So a crucial function of the nephrons is to keep the solute levels in the body at 300 milliosmoles. This is actually the normal number for blood plasma, for the interstitial fluid in all other parts of the body, and for most of the fluids of the body, including the fluids of the cortex. So 300 milliosmoles is where we're actually going to aim. So the countercurrent mechanism will retain this osmotic gradient going from the cortex into the deep medulla, and we'll talk about how it's established, and we'll also talk about why it's important. But there are a lot of details to it, so please pay attention. If you need to listen to this lecture many times, please do so, because all the little details are important, but kind of the big picture will only start to suss out as you have repeated, as you have repeated exposure to the information. So as fluids enter into the medulla, they are at 300 milliosmoles. And the proximal medulla is at about 300, 400 milliosmoles. Deepest inside of the medulla, again, is at 1200. The first thing we're going to discuss is called the vasa recta. Vasa meaning blood vessels, recta meaning up and down. The vasa recta is the particular set of peritubular capillaries that specifically surround the nephron loop, or the loop of Henle. So the vasa recta is this blood vessel system that, as you can see, dips down into the medulla. If we take another look at this, what happens with a nephron is that you have the glomerulus, the PCT and the DCT are going to basically be present out here in the cortex. And then you're going to have the nephron loop that's going to dip down into the medulla, into this salty content here. So the nephron loop dips down into the salty content, and so does the vasa recta. The vasa recta is called the countercurrent exchanger. So in the deep medulla, blood freely exchanges water and sodium with the interstitial fluid. It's basically a very porous blood vascular system. So if we were just to take out all the convolutions and rather than just show it as a descending and ascending tube, what you'll notice is that the salt content of the vasa recta, just look at the numbers inside of the blood, they very closely follow the surrounding tissues. There's a little bit of a lag there, so it's 400 milliosmoles inside of the blood here, and which is not quite exactly matching, but basically the salt is able to flow in, water is able to flow out freely, and then as it goes out, water is able to flow in and salt is able to flow out freely. So it's basically a very permeable blood vessel. So why do we have blood vessels here? Well, the deep medulla is like most tissues of the body. It requires oxygen. It requires nutrients, glucose, and it requires the ability to get rid of waste products. So the purpose of this particular unusual capillary bed is just the same as any other capillary bed. Basically bring oxygen into the region, carry carbon dioxide out. Okay, so in the deep medulla, blood freely exchanges water and sodium ions with the interstitial fluid. When you have the descending limb entering the deep medulla, it loses water, it takes on salt, and the osmolarity will increase. And this makes it basically isotonic or isoosmotic with the surrounding tissues. In the ascending limb of the vasa recta, the blood is going to gain water, it's going to lose salt, and so its osmolarity will go down. And that also, again, makes it isotonic or isoosmotic with the surrounding tissues, the same salt content. So the vasa recta does not cause the salt gradient, but it does allow it to be maintained. In other words, it does not bring extra salt into the region or take any extra salt out of the region. It just basically allows the interstitial fluid to be maintained as it is. There are a few different solutions that we're concerned about as we might make our way through the deep medulla. Let's take a look at this picture here. There is the blood supply that we just took a look at, the vasa recta, that dips down and comes back out of the deep medulla. There's also the interstitial fluid that is going to have this specific gradient. And by the way, the body does not like gradients, so it's always pushing for this salt content to be present, and it has a bunch of mechanisms in place to make sure that it maintains it. However, it's always fighting against the idea that the body likes equilibrium, and so it's always fighting against the gradients. And then the third fluid that is present in the deep medulla is the filtrate that is inside of the nephron loop. So we're going to be discussing each of those three things. Before we go on with this video, make sure that you understand all three of those things. That you understand the filtrate is inside of the nephron loop, which is part of the renal tubule system, that the blood is inside of the vasa recta, and that surrounding both is interstitial fluid. So we are concerned with the salt content in all three of these areas. All right, for this you want to just go ahead and take a look at the picture that you have. If you are really good at making sketches, you may want to just go ahead and make the sketch. The glomerulus is a tuft of capillaries inside of a glomerular capsule. Just a little refresher here. Here's the glomerular capsule, which is the first part of the renal tubule system. The renal tubule system then is followed by the PCT, the proximal convoluted tubule here, followed by the loop of Henle, or the nephron loop here, followed by the DCT, the distal convoluted tubule here. And then this, of course, is the collecting duct, which is going to be collecting filtrate left over from from several different nephrons at once. Okay, so the first thing that's going to happen is that the descending limb of the nephron loop is impermeable to solutes. It doesn't allow them in or out, but it is permeable to water. So water leaves the tubule due to osmosis. Remember, this is dipping down into a salty content, so it's very easy for water just to leave and enter the interstitial fluid where it is saltier. Remember, the numbers aren't written in here, but you might want to just go ahead and write in, 300 up here and then like 1200 down below here, just to remind yourself that the numbers change significantly here. So as you dip down into saltier contents, since the tube is permeable to water, it allows water to just leave and enter the salty atmosphere there. As a result, the filtrate inside of the nephron loop becomes very concentrated. As you dip down to this elbow at the lowest part of the nephron loop, it's going to be at about 1200 milliosmoles, which is isoosmotic with the surrounding interstitial fluid. The ascending loop then, as we dip back out of the deep medulla, the ascending loop is going to be selectively permeable to salts, but not to water. So we already established that the filtrate inside of here is very, very salty. So as you leave out of here, water isn't going to be able to leave or enter. However, you are going to have the ability to release salts. So the thin segment of the ascending limb of the nephron loop, will release sodium ions and chloride ions passively, and then eventually the thick segment has pumps that actually allow the sodium ions and chloride ions to be actually pumped out. So the filtrate inside of this nephron loop actually becomes quite hypotonic or hypoosmotic, and as you enter back into, up here is the cortex, of course, which we already established is at about 300 milliosmoles, but not inside of the renal tubule here. The DCT, the distal convoluted tubule here, is going to have filtrate that as it enters into the DCT is going to be quite hypoosmotic. It's going to be at about 100 milliosmoles. So it basically is lacking a lot of salt at this point because it has been both released and pumped out. So this contributes to the high osmolarity of the interstitial in the fluid in the medulla. These two steps, the release of sodium and chloride ions in the thin segment, and then pumping out sodium ions and chloride ions in the thick part, means that the deep medulla remains salty. It also it causes the filtrate inside of the tubule to become very dilute or very low in salt. So again, the number that I would use here as an example would be 100 milliosmoles. So it's actually hypoosmotic to the surrounding tissues, which we already said were 300. Okay, so the filtrate starts out as it enters into the nephron loop, it starts out isoosmotic with the interstitial fluid at 300 milliosmoles. As it passes through the deep medulla, it continues to be isoosmotic with the surrounding interstitial fluid, so it dips down to about 1200 milliosmoles, and you can look at the details why. It's because it's released a lot of water. And then this will be isoosmotic to the deep medulla, but it will be hyperosmotic or hypertonic to most of the body's fluids, including blood plasma and most of the interstitial fluid of the body. The filtrate itself becomes hypoosmotic, and we already established, as it enters the DCT, it's going to be at about 100 milliosmoles. Okay, the last thing to cover then, now that we've covered what happens in the nephron loop and the DCT, are the collecting ducts. Now, the collecting ducts are permeable to urea, and it exits the duct and enters the interstitial fluid. The interstitial fluid is generally kept low in urea, and so this is one of the things that actually establishes sort of a concentration gradient inside of the deep medulla, that you have this urea content there. Now, the other thing that's going to happen in the collecting ducts is that you have a choice. You can either release water back into the body. Remember that everything inside of the renal tubules is called filtrate. Filtrate is called future urine for a reason. If it makes its way all the way through the renal tubule system, PCT, nephron loop, DCT, and then all the way down through the collecting duct, it will become urine. So it's destined to leave the body. Anything that leaves the renal tubule system, including the collecting duct, anything that leaves the collecting duct, re-enters the body. This is considered the reabsorption of product back into the body. So when I say that the presence of ADH causes the release of water, what it actually means is it causes the reabsorption of water. So what is ADH? ADH is anti-diuretic hormone. It's released by the posterior pituitary gland, and it causes aquaporins to be placed in the walls of the collecting ducts. So when you don't have ADH present, you don't have any aquaporins, and the water remains inside of the collecting duct and does leave as water. One of the reasons you might not have ADH present is that you drink lots of water and you just want to let go of a lot of it. But one of the reasons you might not have ADH present is that you may have taken some kind of a diuretic that actually fights against the role of ADH, which is anti-diuretic hormone. So diuretics include alcohol, of course, that's the biggie, but a lot of caffeinated drinks are also considered diuretics. Okay, all right, so remember, it's what we've been talking about all along. Inside of the deep medulla, it is very salty, it is very highly hyperosmotic. Having that grand salt content there allows water to easily leave out of the collecting ducts, because remember, at this point, this is a very watery filtrate. It is basically 100 milliosmoles. It's incredibly hypoosmotic to anything, much less hypoosmotic to the deep medulla, which is at about 1200 milliosmoles. So it's super easy. If you put aquaporins in the walls of the collecting duct, it's very easy for water just to leave through the process of osmosis and enter into that very salty content. So why is it salty? It is salty because sodium ions and chloride ions are both released and pumped out in the ascending segment of the nephron loop. Additionally, the urea that is released through the collecting ducts helps to establish a concentration gradient in the deep medulla. So that is the why. That is how it gets that way. The reason why it is that way is just so that in this fourth step, you're able to release water back into the body if it is the case that you want to release water back into the body.